So thank you so much for this opportunity. This is a really great initiative, and I'm embarrassed that it wasn't on my radar before now, but I'll certainly be looking through your back catalogue of talks. I'm going to describe our recent paper, which was published about 6 weeks ago in Science Immunology. I won't go and do a deep dive into the data because there isn't time, and it's all in the paper. But if anyone has issues accessing it, please drop me an email and I'll see if I can help. The paper centers on the role of TFH cells in the germinal center, and Ava's just done a beautiful job of introducing the germinal center for us. This is an image of a lymph node from a mouse that's been immunized, and I've highlighted the germinal center here. And what you can see is two zones. So we've got the dark zone here, where the B cells are rapidly proliferating, and the light zone, where you will find T cells. Here's my cartoon of the germinal center. So the B cells are present in the light zone, and there they receive help from T follicular helper cells. These are T cells in the follicle that are there to help the B cells. Once they've received that help, they move to the dark zone and undergo this rapid proliferation and also somatically hypermutate their B cell receptors. And then they go back to the light zone, and those good B cell receptors are selected by their interaction with T cells. Also in the germinal center, we have T follicular regulatory cells. They're not as numerous as the TFH cells, but they play an opposing role. So they are there to dampen the germinal center response. We know a lot about the role of TFH cells in the development of the germinal center, but we don't know very much about their role within the germinal center, what happens in an established germinal center. We assume a lot of things based on what we know about their role in the development of the germinal center. And the work has really been hindered because we don't have any good models where just TFH cells are targeted by, say, CRE. And this is because they're transcriptionally very similar to a lot of T effector cells and very similar to TFR cells. And what Michelle and I set out to do quite a few years ago now was to try and find a model system where we could delete TFH cells in an established germinal center so that we could address some of these questions. We had a look at using IL-21 as a potential gene to induce CRE because IL-21 isn't produced by a lot of cells. It's mainly produced by TFH cells and Th17 cells. And in our animal house, we're fortuitous in some ways in that the microbiome of our mice isn't conducive to Th17 cell development. So we thought if we could get a fate mapper mouse that would mark IL-21, we had a way of maybe targeting just TFH cells. Fortunately, there was a fate mapper mouse for IL-21 made over 10 years ago now, and it was originally made to fate map Th17 cells. The way this works is that it will fate map any cell that expresses the IL-21 gene. So that the ROSA locus, which contains the RFP, expresses RFP continuously. So once the cell has expressed IL-21 gene, it is permanently marked with RFP. So we imported these mice and we gave them an immunization. And throughout this talk, I will swap between MPKLH immunizations and flu infections. And we've used both to characterize these mice and also the phenotypes that we find. So when we did this, we put the cells through flow and we did some t-SNE. So here you can see just a really broad kind of clustering. So we've got CD4s, CD8s, and then we've also put our FOXP3 cells, which would be the TFR cells and the other T regulatory cells, and the CD19 cells. And then if you look at the expression of RFP, which is marking IL-21 fate map cells, you can see there's a very tiny proportion within the CD4 gate. It co-localizes with CXCR5 expressing cells, and there's no expression within that FOXP3 positive population. So it looked very hopeful that maybe this was marking our TFH cells. If we look more in detail, we can take our activated CD4 cells and mark them with CXCR5 and PD1. And this is a way of demarking different types of TFH cells throughout their differentiation. So we have the non-TFH cells, which don't express either marker, the pre-TFH cells, which are a precursor population, which have intermediate levels, and then those that express high levels of both CXCR5 and PD1, and these mark our TFH and our TFR cells. If we look at the non-TFH cells, we can see that less than 5% of these cells are expressing RFP, so less than 5% are IL-21 fate mapped. This goes to about 40 to 50% in the pre-TFH cells, and over 90% of our TFH-TFR cells are positive for IL-21 fate mapping. And that's shown here in this graph. We can further divide these TFH cells into germinal center resident TFH cells and TFR cells. And this is what we've done in this flow plot here. So we've made a slightly larger gate for our TFH cells. And then we've looked at FOXP3 expression relative to IL-21 fate mapping. And what you can see is that there is no co-localization. So it appeared that our IL-21 fate mapping was restricted just to our FOXP3 negative TFH cells and not the FOXP3 positive TFR cells. There is data in the literature to suggest that when you do a prime boost model of germinal centers and look at TFH cells, they will acquire FOXP3 expression. So this was something we wanted to check within our fate mapping cells, although this is a primary immunization, so it does differ. But what we were really pleased to see was there was no co-localization of FOXP3 with IL-21 throughout the entire course of the germinal center response up to day 18, and we have looked further. So whilst the number of FOXP3 positive cells are increasing, the number of those that express the RFP, which is just kind of around about 4%, 4 to 2%, so background levels, remains constant. So we do think that our IL-21 is very selective in its fate mapping of TFH cells and a portion of the precursor TFH population. Of course, you want to look at the imaging and to see these in situ. So this is an image of a germinal center from an immunized mouse at about day 14. And what you can see here is these lovely red cells with the yellow halo. So the red is marking the RFP and the yellow is the PD1. So these are our TFH cells, and as expected, they are in the light zone and a little bit in the B cell follicle. So fitting with our flow data. So now we want to see if we can use these mice to delete these cells. And so what we did was we introduced the diphtheria toxin receptor transgene into these mice. What this means is that if a cell expresses their IL-21 Cre, that cell will, as well as expressing RFP, will express the diphtheria toxin receptor. And so you can then administer diphtheria toxin to these mice at a time of your choosing, and that will delete those TFH cells, so take them away. So we tried this out. We immunized our mice with MPKLH, and we let that germinal center grow and develop as it normally would. And at its peak, at about day 8 and 9, we then administered diphtheria toxin to these mice and looked to see what had happened to our TFH cells the day later. So here we have our control mice, which are expressing RFP, but do not express the diphtheria toxin receptor, on our experimental mice that express the diphtheria toxin receptor. And what you can see is that we have a lovely population of TFH cells in our control mice, but it's completely gone in our experimental animals. And this is quantitated here. We can further subdivide and we can look at those germinal center TFH cells and see that they are also gone. But nicely, the vast majority of CD4 cells are unaffected by this ablation. And we can also see that our precursor TFH cells are slightly affected, indicative of the fact that only about 40 to 50% of them are fate mapped. So we had a lot of ideas about what might happen to the germinal center if we took away TFH cells, but we thought we would ask ChatGPT. So I put this question to ChatGPT about 1.5 years ago. I wonder how it would change now with its answers. But what it said was that there would be decreased germinal center formation and maintenance. And that made sense. We know that TFH cells are important for the development of the germinal center, so it's likely they're important for their maintenance. It said there would be increased apoptosis of B cells, and this is because there are papers that suggest that TFH cells give a survival signal to the germinal center B cells. And so if you take away the TFH cells, there's likely to be more death. It also said there would be a loss of B cell selection and affinity maturation. And this also made sense because the T cells are there to select for the good B cell receptor carrying germinal center B cells. And so if you take them away, that selection event will probably be affected. And then it said there would be weaker long-term immunity. We know that TFH cells are important for promoting memory B cells and long-lived plasma cells. So if we take them away, what will happen to that? What will happen to the memory response if you come back with that same antigen later? So we set about testing each of these. And the first one was to look at the formation of the germinal center. Well, not the formation, but its maintenance because it's already formed. And so here we've used the same setting that we did last time. So we've given them mice MPKLH, then diphtheria toxin after the germinal center is formed, and then looked at that structure on day 10. And so here we're looking at the germinal center B cells, which are marked by Ki67 and BCL6 expression. And you can see we've got a nice population in our control mice that don't express diphtheria toxin receptor, but it's diminished in our experimental mice, and that's shown here. But what you can see is that the B cells aren't completely gone. There are still quite a few there, so it's not an absolute reduction. If you look by imaging, you can see that the germinal center is smaller. So here's the merged phenotype, the merged images. So this is our control mice and then our experimental animals. And we can enumerate the different cells in here. So here we're just enumerating the R21 fate mapped cells in the germinal center. And you can see that in our experimental animals, they are gone as we had seen by flow, but we're now confirming it by imaging. But the number of germinal centers per mice in each mouse didn't really change hugely. And the difference in size was not really significant unless you looked particularly at the size of the dark zone. So this is the area where the germinal center B cells are going through that rapid proliferation. And the light zone size was very similar. So it seemed that the dark zone was particularly affected, maybe because there was a proliferative defect when TFH cells were taken away. We then went to look at what happens to the survival of these germinal center B cells. If we take away the TFH cells, is there increased apoptosis? So we went a day later this time because we know that actually the germinal center response, the germinal center B cells decrease in number after day 10. And so day 11 seemed a better time point to look at. And we looked at caspase-3 expression, so as a marker of apoptosis in both the dark zone and the light zone germinal center B cells. And there was no significant increase in caspase expression in either the dark zone or the light zone cells. And we've done this at various different time points, and we don't see any significant effect. So is there a proliferative defect? Is that the reason why the germinal center numbers are going down and why we have a reduced size of the dark zone? So we gave the mice EDU. EDU is incorporated into the DNA and so marks proliferation. And what you can see in the flow plot here, which is gated on germinal center B cells, is that whilst we have a good portion of EDU expressing cells in our control mice, it's significantly down in the experimental animals. And you can see that here. It's about a 40 to 50% reduction. This makes sense to us because TFH cells induce a signal to germinal center B cells that drives MYC expression. And we checked this in our system by using a MYC reporter mouse. And what we can see here is that in our control mice, we have good induction of MYC in the germinal center B cells, but it's completely lacking in the experimental animals where the TFH cells had been depleted. So it looks like we're affecting proliferation of germinal center B cells, and that is affecting the size of the germinal center. But this is just the early time points after depletion. What happens next? We weren't really sure. Was the germinal center going to crash further? Was it going to maintain that smaller size? Or would it reboot in some way and get back to the size that it was beforehand? And so what we did was a kinetic experiment. So the mice are immunized and then given diphtheria toxin at the peak of the germinal center response time. And then we looked at different time points following this. And I'm just showing here the number or proportion of germinal center B cells. So in red, we've got our control animals, and in gray, we've got our diphtheria toxin receptor expressing animals. So these are the ones where the TFH cells will be depleted. If we look at day 8, prior to diphtheria toxin administration, then there's no difference. We see the drop at day 10 that we'd seen previously, a further drop at day 14, but surprisingly, by day 18, the numbers were back to normal. If we look at an image of a day 18 germinal center, what you can see is that this is the control germinal center here, and this is the one where the TFH cells have been depleted. The sizes come back to a relatively, it's more or less the same size as the control. But what you should also be able to see is the TFH cells have returned. So you can see these red cells with the yellow halo. So that's RFP with PD-1 expression. And we can enumerate that here in the same way, but we've just imaged here on the germinal centers rather than flow. And so you have normal numbers at day 8, completely gone by day 10, and then it picks up again at day 14, further at day 18. But what we can say is that this number never gets back to control levels. So whereas the germinal center B cells at day 18 had reached the normal levels of a control mouse, if you take away TFH cells, the numbers never return to that of a control mouse. And we think this probably has some important implications for memory responses, which I'll come on to. So we've lost our germinal center B cells, we've lost our TFH cells, but they're coming back. But they're coming back in different proportions. So there are more germinal center B cells coming back than TFH cells. How is this going to affect affinity maturation? Because it's very obvious that there's a clear link between the state of affinity maturation and the ratio of TFH cells to germinal center B cells. The dogma is that you must have fewer TFH cells relative to germinal center B cells so that there is competition, so that those germinal center B cells have to compete for TFH cell help. So we looked at this, and we looked at day 14, which is the time point where we've got the least germinal center B cells. It's where the effect is at its greatest in terms of the germinal center. And so here we're just showing the germinal center B cells, and you can see that in the experimental mice that have had the TFH cells removed, it's significantly lower. And because we're using MPKLH, we could look at the antigen-specific germinal center B cells this time, and we can see they're significantly down. We did the ELISAs, very similar to what Ava just showed, but we also did some single cell sequencing on the B cells from these germinal centers to allow us to identify the high affinity mutants. So in the MPKLH model, when a B cell becomes high affinity, it mutates its tryptophan to a leucine. And so you can look at that and analyze it. And what you can see is that in our control mice, about 50% of the B cells have become high affinity. Surprisingly, in the experimental mice, that has increased to about 75%. We think this is probably because there is a reduced ratio of germinal center TFH to germinal center B cells. And so there is greater competition for that TFH cell help. But what happens after this time point? So if we look at day 21, when we know the germinal center B cell numbers have come back to normal, but the TFH cells are still depleted, and that's shown here, we can see that actually the proportion of B cells with the high affinity mutation is actually equal between the control mice and the experimental mice. So affinity maturation seems to have normalized again. And this is despite the fact that there is still this reduced ratio of TFH to germinal center B cells. It's not as reduced as it was at day 14, but it's certainly still there. And finally, we wanted to look at the long-term immunity. What happens to memory responses? So here, I'm just going to show work that we've done in flu, because here we could mark the antigen-specific cells in the CD4 population by using tetramers. So here we've given our mice flu infection, and then we've given them diphtheria toxin at the peak of the germinal center response, so days 8 and 9. And then we've looked 6 weeks later to see what are the proportions of antigen-specific T and B cells. How has disrupting that germinal center all those weeks ago affected these cells? And what you can see is that there's a clear reduction in the proportion and the number of MP-specific CD4 cells. So there is a significant reduction in the memory T cells that we believe have come out of this germinal center, because all these cells are fate mapped. And this makes sense because we know that the TFH cell population never gets back to normal numbers after we've depleted at that time point. In contrast, our antigen-specific B cells show no defect. And this fits with what we saw with our germinal center B cells, that they seem to recover in terms of their numbers after this disruption of the germinal center. So we've got a normal proportion of MP-specific B cells and also a normal number. So then we asked how this would affect these mice if we gave them a reinfection with flu. So we've given them flu, we've deleted the germinal center at days 8 and 9, and then 5 weeks later, we've given them a boost to look at their recall responses. So here, if we look at the TFH cells, we can see we've got this lovely population in our control mice, but it's not really there in our experimental animals. And bearing in mind, we're doing this very early now, we're doing this at day 7 after the boost. This is really the memory cells that have a quicker and better activation. And we can enumerate that here, and we can see they're significantly down. And what we also see is that the germinal center B cells are significantly down as well, which fits with the TFH cells helping those germinal center B cells in that second response. So finally, we want to ask, what is the role of these TFH cells that have come out of that germinal center? So here we've given the mice flu, and we've let the primary germinal center and that primary immune reaction just occur without any disruption at all. And then just prior to the boost, we have given the mice diphtheria toxin. So this will take away those TF8, those CD4 T cells that have been fate mapped, those ones that have participated in the germinal center. And what we see here is that we have a nice TFH response in our control mice, but it's very reduced in our experimental animals, and that's enumerated here. And as we saw before, this has a knock-on effect in terms of the germinal center B cells, where there is a significant reduction as well. So it appears that the export of TFH cells from that primary germinal center are very important for our secondary recall responses. So I'm going to summarize the work here. What we were looking at was how the germinal center is affected when you take TFH cells away. What we've done is allow a germinal center to form and then to delete the TFH cells at the peak of that germinal center. What we see is a pausing of the germinal center B cells at this time. So they remain there, they seem to survive, but they stop proliferating. until we get an influx of fresh TFH cells coming into that germinal center. And this seems to reboost those germinal center B cells to start proliferating again until they get to the capacity that they should. However, the T cells never get back to the original numbers, and so the pool of TFH cells within that germinal center remains reduced. It never fully recovers. And so when you reinfect or re-immunize or give a secondary challenge 7 weeks later, the recall germinal center response is impacted. So whilst many reviews on germinal centers say that the main role is to produce antibody secreting cells and memory B cells, we would argue that actually the production of memory TFH cells is also a sentient role of the germinal center. We think this pausing of the germinal center response might be an evolutionary strategy. So a lot of lower invertebrates and some embryos, some mammals during embryogenesis can go through a state of diapause, which is a way of pausing the metabolism and the activities of that cell in conditions of stress or loss of help. And we think that this is possibly what's going on within our germinal centers, so that when there are stressful conditions, the germinal center B cells pause until they can get the factors to get them back into action. And so they can survive that current infection, but it comes at expense for preparedness for their next infection. I think it shows that there is a vulnerability window in the germinal center reaction, so that if TFH cells are disrupted by whatever means during this peak of the germinal center, then that can have implications for recall responses. We know that factors such as stress, inflammation, co-infection, and aging can all affect TFH cells and divert them away from that TFH pathway. And so this is likely to have impacts in the formation of immune memory. I will stop there and just acknowledge the great team that has really helped with this whole paper. It was a huge team effort. The people I would really like to signal out are Helena, Jess, and Emily. Here's Helena here, and here's Jess. Emily's now doing a PhD at the LMB, but they've all made really big contributions to this work and all came in as this was kind of their first time in a lab, so it's been really